which species is the vector? Okay. First of all, there is no such thing as the vaccine. A flu shot will do nothing for measles. A measles shot will do nothing for tetanus. Tetanus does nothing for COVID. Each vaccine is only good for one disease. You have to have the right vaccine. So now we're going to be talking about mosquitoes and disease. You click over here. Mosquitoes transmit a whole bunch of different pathogens. They're responsible for about 250 million cases of disease every single year. And this results in about 700,000 deaths every single year. Some of the common diseases.
Mike, your slides are not progressing.
You're kidding. Back to that again. Is that working or not?
No.
Hold on one second. Let's get. Okay.
That did.
Okay, just for the fun of it. I'm going to do this the old-fashioned way. Forget the projection one. So you're seeing right now vector-borne diseases, 250 million?
Yes.
We'll just go down that way.
Okay.
And I may cheat a little bit over here. That works. Yeah. Okay. See what happens. So some of the common diseases include malaria, yellow fever, dengue, Japanese encephalitis, West Nile. You've all heard of most of these diseases, so I'm going to skip it. Well, what makes a vector a vector? And the important part of this, and why is that important? And that's my presentation today. Why is it important to know which species is a vector and some of the factors that affect what making it a vector? A mosquito is not a mosquito. There's more than 3,500 different species of mosquitoes. A lot of mosquitoes are annoying biters. Very, very few of them transmit disease. And despite what you hear all the time, there is no such thing as mosquito control. There's no such thing as the vaccine. Your control is all directed at specific species of mosquitoes. Is it a day-biting mosquito? Is it a night-biting mosquito? Is it a salt marsh mosquito? Is it a mosquito in a flower vase? Your control method is dependent on the different type of mosquito. And unfortunately, controlling the wrong mosquito species may not help reduce pathogen transmission. And there's actually some well-documented cases where doing really good mosquito control killed a whole bunch of people from disease. For West Nile virus, doing really, really good control in the Midwest in a non-study, but it really was a study. They did really good control. They controlled Aedes vexans, annoying biting mosquito, their principal pest, and they killed a whole bunch of people. There was more than 3 times as many deaths per 1,000 people where they did the good control than where they did regular control. So you need to control the right species. So we can control the right species so that we can reduce disease and not just annoyance. Well, how do we incriminate a vector? Well, people are collecting mosquitoes looking for disease, for pathogens all the time. If you find a particular pathogen in a mosquito species, in a particular mosquito species, there's an association in nature between that species and that virus. So that's a possibility there, but it doesn't mean that it is a vector. Demonstrate that the arthropod feeds on the right host. If this is a disease like dengue or yellow fever, which is a human disease, human mosquito, parts of South America or Africa, monkeys get involved, but monkeys are just ugly humans. So it's primate mosquito. If this mosquito feeds primarily on birds, it's not going to be important. Demonstrate that the arthropod is active at the right time of year. In this part of the United States, West Nile is active in the fall. If this mosquito only occurs in the early spring and then disappears, it's not going to be involved in human transmission of West Nile virus. And finally, to me, the most important part of this is the demonstration in the laboratory that that particular species is able to become infected and can transmit the pathogen by bite, i.e. vector competent. It is a competent vector that can transmit it. There's lots of mosquitoes that get infected in nature. In studies that I've done where you've got lots of infected mosquitoes collected in the field. You bring them into the laboratory, they can't transmit the virus. They're incompetent. So they're not, even though they're associated with the virus in the field, they get exposed to virus, but they can never transmit the virus. Therefore, they're not dangerous. Well, vector competence versus vectorial capacity. I was trained in vector competence, and some of the people I worked with after I got my doctorate convinced me that vector competence is nice, but vectorial capacity is much more important. Well, vector competence is, can this mosquito transmit this pathogen? If 100 mosquitoes get exposed to the virus, what percent of them will be able to transmit it? So the higher that number, the higher the vector competence. Is it a competent vector? Vectorial capacity is, how important is this species in transmitting the virus? Not how efficient it is, how important it is. You can have mosquitoes which are extremely efficient, but they feed on the wrong animals. Therefore, they're great in the laboratory, but they're meaningless in the real world. So we have to put a whole bunch of factors together. Well, how do we determine vector competence? Well, one, can the virus infect the mosquito? So what I would often do is take bi-isolated virus from Peru, Ecuador, Costa Rica. I have a field-collected virus. If it's normally a bird virus, I take a baby chicken, inoculate baby chickens with the virus, and do viremia profile to know when the birds were viremic. If it was a mammal virus, inoculate hamsters with it. So I would know when that particular infected animal would have virus in its blood. I then feed mosquitoes on it and sample at various time periods. Well, if we have a mosquito that is just a blood meal full of virus, and you test this mosquito, it's full of virus. However, if there are no receptors in the midgut, the midgut does not become infected, the blood meal gets digested, excreted, and it's gone, and you're back to an uninfected mosquito. So you can't test the mosquito right after it took a blood meal because it's full of virus, whether it's infected or not. If there's no infection in the mosquito is said to have a midgut infection barrier. If the midgut gets infected, the virus grows in the mosquito. It's infected. It's infected for life. It's full of virus. But the virus is stuck only in the midgut. This is sort of like a polio infection in a child where the polio virus does not get out of the gut. Not serious illness. Don't worry about it. These mosquitoes are infected, but they cannot transmit it. They're said to have a midgut infection barrier. If the virus gets out of the midgut, it goes all over the whole body in like one day, really fast. There's virus everywhere, and there's about 100 times more virus now than there was in the midgut alone. It's full of virus. However, in order to transmit, the virus needs to get into the salivary glands. And pointer. Salivary glands are right over here. A little blue thing here. Those are the salivary glands. If the virus does, there's a membrane around the salivary glands, like there's a membrane around the midgut. If the virus can't get through that membrane, lots and lots of virus, and this mosquito has what's called a salivary gland barrier. They can't transmit. But if the virus gets into the salivary gland, into the saliva, if that mosquito so much as probes for 3 seconds, the virus is in the saliva and is ejected. Remember, when a mosquito wants to take a blood meal, it lands, it takes its proboscis, sticks it into the skin, and the first thing it does is spit out a bunch of saliva to stop blood clotting so it can find the blood vessel. Within seconds, and we're talking a couple of seconds, that mosquito has salivated, has transmitted the virus. It does not need to find blood to transmit the virus. This is I'm going to skip this. This is how I do all the studies. I feed them on an animal, separate the engorged ones, and then refeed them to see if they transmit, and grind up mosquitoes to see if they're infected, but who cares? Okay, vectorial capacity is how important this mosquito is in transmitting the virus. And there are various factors that affect vectorial capacity. And these include vector factors, actually, what's the word here? Virus factors, vector factors, host factors, and the environment. All of these influence whether or not that particular species of mosquito is an important vector and needs to be controlled. If we look at the virus factors, does this particular strain of this particular virus produce a high enough viremia to infect the mosquito? If it produces a high viremia, can it grow in that species of mosquito? Is it a pathogenic virus to animals, pathogenic virus to humans, i.e., does it cause disease? If it doesn't cause disease, who cares? There's a whole bunch of viruses out there that do absolutely no harm whatsoever. We ignore them because who cares? If they cause disease, we worry about them. If they are virulent, i.e., they cause severe disease, we worry about them a lot more. So these are all virus factors. The vector factors. Is it a competent vector? Can this mosquito transmit that virus? You can have a mosquito which is super competent, unbelievably efficient, and it's on the endangered species list. Who cares? There's not enough of them out there to do any damage. If it's a more common species, then it's much more dangerous. What vertebrates does it feed on? As I mentioned, if you have a mosquito that feeds on birds, it's not going to transmit dengue or chikungunya or Zika. When does it feed? Where does it feed? Some mosquitoes readily go inside houses, others do not. Some mosquitoes feed only during the day, some only feed at night. Short digression, back about 50 years ago, it was a severe hurricane in the Caribbean, destroyed a lot of housing. It was a dengue outbreak, and a bunch of churches got together to say, there's a dengue outbreak, people's houses were destroyed, people are living in huts, essentially. We need to stop the dengue outbreak. Donate money, and we will buy treated bed nets to stop this outbreak. And they got a huge amount of money, put in thousands upon thousands of treated bed nets into those villages. They did not prevent a single case of dengue. Dengue is transmitted by a mosquito that only bites midday. So unless you're sleeping in your bed net at noon, it was worthless. However, people were using the bed nets. It greatly reduced the malaria because the malaria mosquito bites at night. So on your control thing, you have to know when it feeds. Does it go inside the house, outside the house? Flight range. If you're talking Aedes aegypti, the dengue mosquito, if you're talking 150 yards, a little bit over 100 meters, man, that's a long way for that mosquito to fly. It will not go that far in its life. If you're talking Aedes tenurensis, and you're on an oil rig 15 miles off the coast in the Caribbean, they'll come get you every night. They'll fly up to 20 miles a night to get 30 kilometers a night to get a blood meal. So if you do control, you kill every single mosquito within a 5-mile radius of your house. You kill every last one. Don't worry about Aegypti because it'll take them weeks to get back in there again. Tenurensis will be back tomorrow. They don't care. They'll fly 20 miles. So how far, what kind of spraying do you need to do? Where do you need to do the spraying? Vertebrate factors. Again, does this vertebrate produce a viremia? Some West Nile is a bird virus. You put it into adult chicken or duck, the virus goes, whoa, where am I? I'm dead. And the virus does almost no harm and produces no viremia. The virus is dead. You put it into a crow, it produces a trillion virus particles per gram of brain. It's unbelievably high titers, 10 to the 12th. So a bird is not a mosquito is not a mosquito, and a bird is not a bird. Depends on what species of bird it is. How common are they? When West Nile broke out, and crows were really, really important. There was a Hawaiian species of crow they were really worried about. Could this crow, because it's a Hawaiian crow, be involved in transmitting West Nile? Well, there are only two specimens of that bird still alive. It's extinct now because they died of old age. But no, if there are two birds in the whole world, they're not going to be important. It has to be common. If it's not common, it's not going to be important. Does it get sick? This is important for several reasons. One, if they don't get sick, we don't worry about it. The other is if you are alive and well, or a dog or a cow is alive and well, and a pest comes to bite it, it does whatever it can to shake the beastie off. If it is really sick, it just goes, and all the mosquitoes in the world will feed on it. So if it gets sick, it's more likely to get fed on, which means it's more likely to infect a mosquito. Antibody prevalence, otherwise known as herd immunity. If you've had, West Nile has outbreaks every 3 to 4 years, because you have an outbreak, virtually every bird is infected. The next year, all the young birds had mothers with antibodies, so they have antibody, you get very little transmission. And for several years, an infected mosquito bites an immune bird, virus is dead, you don't get transmission. When the antibody level gets low enough, all of a sudden the forest fire hits again, and it's explosive. It's just like a fire in a forest. If you've had recent fires, it's hard to have a big forest fire. If it's been 20 years since it's been a forest fire, you're much more likely to have a big fire. And which mosquitoes feed on it? Again, different mosquitoes feed on different things. So for this animal to be important, mosquitoes have to feed on it. And finally, environmental factors. Temperature is very important. If it's cool out, mosquitoes their temperature is ambient temperature. So whatever the temperature where they are is, that's the temperature of their body. Virus growth is a chemical reaction. If it's really cold, the virus grows really slowly. And even after 30 days, the virus has not made it all the way to the salivary glands. Mosquito's not going to last 30 days, but the virus still can't be transmitted. So therefore, you get no transmission. If you're up about 30 degrees, man, that virus goes right through the mosquito. It is 28 to 30 degrees is for most viruses, is the optimum temperature for more efficient transmission. You get up to 35, we're talking Celsius, up to 35 Celsius, man, that virus goes really fast. Unfortunately, it's so hot the mosquito dies. Because mosquito can't stand that. So temperatures of about 28 to 30 Celsius are the most dangerous temperature for virus transmission. Rainfall. Mosquito larvae develop in water. No rainfall, really dry, no mosquitoes. On the other hand, because there's no rainfall, it's really dry, particularly in the tropics, people have to store water in barrels by their kitchens so they can have water to cook with. That's the water that Aedes aegypti lives in. So it's actually more dangerous if you store water by your kitchen. So having more water means you don't have to store, having more rain, you don't have to store as much water, it's all very complicated. Whatever I say, I can give you an example where it's the opposite. Presence of larval habitat, areas where larvae can develop. And again, larval habitat, is that a salt marsh? High tide flooding? Is that a floodplain where you have a whole flat area, really heavy rain, and this whole area fills with water, and you get a gazillion mosquitoes because the Aedes eggs will live there for 5 years, sitting there. And when that floods, man, every egg for the last 5 years hatches, and you get clouds of vicious mosquitoes coming off. You have mosquitoes that only uh, but lay eggs into little containers holding water, coconut shells. If the coconut shell is this way and it fills with rainwater, the Aedes aegypti eggs in there will hatch and you'll get 100, 500 eggs out of, well, 100, 500 adults out of one stupid coconut shell. Uh, that's the mosquito that transmits dengue, Zika, chikungunya, yellow fever. If you put out, and I know one of my wife's friends had a big flower urn in her back porch, and she had the little saucers underneath it that hold water. So you fill those with water so the plants always have access to water. I went out there in her back porch and looked at it, pointed to all the little tiny worms squiggling in her water. She probably had 500 larvae slash pupae in every one of like the 8 saucers. She had several thousand mosquitoes that she was growing on her back porch. These were almost all Aedes albopictus, the Asian tiger mosquito, which is a vicious biter and one of the principal pests in this area. I helped her today put her flowers on the back porch. She no longer uses those cups. She puts them on a little screen thing, and she has to add water. Now she has to water them more frequently. But she's reduced mosquitoes by 2,000 mosquitoes a week out of her back porch. Larval habitat is critical. Real quickly, an anthraconosis versus zoonosis. And anthraconosis, these are diseases that the source is a human. Dengue, malaria, chikungunya, Zika. These were humans are the source of infection. The mosquito gets the pathogen from a human. In my typical little example here, we have Sally. Sally has dengue. A mosquito, Aedes aegypti, the vector, bites her. Mosquito gets infected. The mosquito can transmit the virus, and he bites a dog. The dog gets infected, produces so little virus that if a mosquito feeds on the dog, it does not become infected. It does produce antibody, so you can monitor dengue in an area by monitoring the dogs, but the virus is dead. If, however, the mosquito bites John, John gets dengue. John produces a high viremia in dengue because that's what this virus does. It's a human virus, and is bit by, why is it like that? Gets bit by Culex pipiens, our typical house mosquito, the principal vector of West Nile virus. The virus goes, whoa, I don't know where I am, and dies because that virus cannot grow in Culex pipiens. So virus is dead. But if it's fed on by Aedes aegypti, it gets infected and bites Sue. And the virus goes round and you get an outbreak. But it has to be the right mosquito that feeds on humans. If that mosquito will bite humans, but bites any other animal, it'll pick up the virus when it feeds on an infected human and then transmit it to an animal where the virus is dead. You don't get an outbreak. You need a mosquito that preferentially feeds on humans.
Mike?
Yes.
Does the virus grow in the mosquito?
Oh yeah. Mosquito ingests a little bit of virus, and I've got them up to titers of more than 1 billion virus particles. If I inoculate the mosquito with 1 virus particle, I'll get 1 million virus particles out.
Wow.
So it grows extremely. If it's the wrong mosquito, I inoculate it with 1,000 virus particles, and I test it a week later. There's no virus.
Yeah.
If it's the right mosquito, and I inoculate it with 1 virus or 1,000 viruses, there's several million virus particles a week later.
Doesn't that kill the mosquito then?
People have said for years that virus infections kill vertebrates but don't harm mosquitoes. I've done a bunch of studies that have found that a virus infection is not good for you. And I don't care whether you're a dog, a cow, a chicken, a human, or a mosquito. We did some really neat studies where I took some mosquitoes, fed them on a viremic animal, waited a couple of days, waited a week, and pulled off the right middle leg from every mosquito. Tested them, and then put each mosquito, numbered them 1 to 50, and put them in 50 little cartons. Tested those 50 legs. Some of the legs were infected, some were not. If the leg was infected, most of those mosquitoes were dead a week later. If the leg was not infected, most of them were still alive a week later. Now, if it has a midgut-only infection, let's go back to polio. You've got a gut infection. It does essentially no harm. Maybe a little diarrhea, but nothing serious. If it gets out of the gut, gets throughout your body, gets into its brain, it really screws up the mosquito. Their fecundity goes way down. Their survival goes down. And I did one other crazy study where I took mosquitoes, fed them on a viremic hamster, waited a week, gave them a place to lay eggs, a whole cage of mosquitoes. Every one of them had fed on that hamster. Took another hamster, anesthetized to put on top of the cage, and waited until just over half of the mosquitoes had taken a blood meal. Removed the hamster, pulled the mosquitoes out, and then tested each mosquito for infection. And I had 3 types of mosquitoes. Mosquitoes that had no virus in them, mosquitoes that had virus in the body but no virus in the legs, i.e. only virus in the midgut. So virus is only affecting the midgut, not much of the mosquito, or viruses in the legs, which means the whole body's infected, including the brain. If they had no virus in them, 75% of those mosquitoes had taken a blood meal. If there was virus in the body, but not in the legs, i.e. the brain was uninfected, 75% had taken a blood meal. If they had virus in the legs, only 25% had taken a blood meal, and 75% had not. Their brain was messed up and they couldn't find blood. Think of it from the virus's point of view. If I'm the mosquito, I'm ready to transmit virus. And I come over and bite. And I sit there and I get a blood meal. Remember, within a few seconds, I've transmitted. I sit on your arm for 2 minutes, I get a full blood meal and fly away. I get 1 transmission. It will take me a week before I digest the blood meal, lay the eggs, and feed again. During that week, a dragonfly is going to eat me. A spider is going to eat me. So I get 1 transmission. If, however, I land, try to find blood, I can't find blood, and the person twitches their arm, I fly away. I land on someone else's arm. I try a second time. I can't find blood. I try a third time, a fourth time. By the 5th time, somebody swats me, or the spider gets me, whatever. I've gotten 4 or 5 transmissions. It is so much more efficient for the virus to disenable the mosquito to find blood. So on one end, if the mosquito is infected, it can't find blood, so it feeds on multiple hosts trying to get that blood meal. On the other end, if the vertebrate is infected, because it is sick, it doesn't have anti-mosquito behavior, and because it has a fever, it's more attractive. So if you take 2 twin animals, put them in the same room, one infected, one uninfected, the mosquitoes just hone in on the infected one and not the uninfected. So on both ends, virus has learned how to greatly enhance transmission. Real quickly now, if you talk to zoonosis, go back up one. A zoonosis, these are diseases where animals other than humans are the principal ones. Eastern equine encephalitis, which is found here. You normally have this transmission cycle going on in Eastern Maryland every single year in the forest, and nobody cares. It does kill birds, but nobody notices a dead bird in the forest. If, however, it gets into pheasants, where someone is growing pheasants or growing emus or something like that, it'll kill them, and you get an explosive outbreak in those birds. That's a financial disaster. And then you get other mosquitoes involved in the transmission cycle. The Culicida melanura, the ones, oops, pointing my fingers doesn't work. The Culicida melanura over here bite birds, so we're not worried about them. However, these other epizootic vectors are more general feeders, and some of them, I've lost the slide over there. Over here on the right, which you can't see because the gene is hiding it, you have a human and a horse. If they get bit, they get Eastern equine encephalitis. They often die. It's a very high mortality rate. It's an evil, evil virus, but they do not produce a viremia. So it's called a dead end host, not because the virus kills them, but because they can't produce enough virus to be transmitted again. Okay, and basically some of the recent introductions we've all heard about: West Nile, chikungunya, Zika. What's next? Rift, Venezuelan equine Japanese encephalitis. I think I've killed up enough time, so I'm just going to stop there and say questions. Anybody have any questions?
It's an awful lot to digest.
Well, I've been playing this game with this stuff for over 40 years, almost 50 years.
And every one of these mosquitoes has its own life history, its own method of reproduction, its own breeding sites, and
It's,
makes it extremely difficult if you don't know how to hone in on the right one.
well, I am one of the world's in this area. I'm one of the world's experts, and I know enough to know that I don't understand it at all. Because every example I give, I can give where that's wrong. I can give examples where where I just said for this other disease, this other mosquito, this other vertebrate, what I said was completely wrong because it does not apply. The scary part is people who have learned a little knowledge and think they understand it, and they know what the answer is. And yes, sometimes they're right
Yeah,
for that guy, but overall, they're wrong.
yeah, we got someone like that in the Pentagon.
We've got that everywhere. So no, I've had fun playing with this and working. I have worked with many technicians over many years. I have learned something from every last technician because they're hands-on doing this stuff and they see stuff that I don't see. So I've had fun, this is to me is a game. I've gotten to travel to Peru, Ecuador, Costa Rica, Belize, Honduras, Trinidad, Mexico, Kenya, Senegal, Liberia, Thailand, Korea, Philippines, Uzbekistan to catch bugs. They paid me to go catch bugs. Idiots. Now,
Hell, I'd volunteer.
the job I had for I worked at USAMR for 35 years. For the first 33 years, I would have paid to have that job. Forget the money. You're sending me to the jungle. And yes, I'm working 18-hour days catching mosquitoes. But in my free time, I'm also picking up any other interesting creature. I got to play with all kinds of amazing things. Yeah, all kinds of just amazingly cool things. Any questions? Any questions online?
I have a question.
Yes.
Albopictus is certainly a big nuisance. Is it anything to worry about in terms of virus?
Yes. Albopictus, the Asian tiger mosquito, for chikungunya in the Indian Ocean outbreak back about 2007, there was this huge outbreak, and it was the principal vector of chikungunya. In the laboratory, it's a much more efficient vector than Aedes aegypti. Aedes aegypti, if you put into a big barn with 100 different animals and one human, most of them are going to find the human and bite the human. They like humans. You put albopictus in the barn, you're going to get a dog blood. They prefer dogs. They like cows, sheep, chickens, birds, humans. They'll eat anything. Around the Indian Ocean, there was not a lot of competition between humans and other. There were no dogs, so they couldn't find dogs. They were biting people. Therefore, they had a big outbreak, and they were the principal vector. There have been several other outbreaks. There was an outbreak of dengue recently in actually Indonesia area. Sorry, and just north of Australia, in Malaysia area, where albopictus was the vector. So yes, albopictus in the laboratory is an efficient vector. It does feed on humans, but it's normally not efficient for an outbreak because it feeds on a human, gets the virus, and transmits to a dog or a cat or a cow. If you look at chikungunya and Zika, both of which are highly efficiently transmitted by albopictus, look at the outbreaks 10 years ago. We had more than 10,000 imported cases. The only locally transmitted cases in the U.S. were southern Florida. There was one chikungunya case last year in New York, so it now has happened. One case. But the only cases up till then were in Florida, Texas-Mexico border, where there were Aedes aegypti, because Albopictus has a higher vector competency. Aegypti has a much higher vectorial capacity. It's more efficient in the real world. Albopictus is more efficient in the laboratory, but Aegypti is more efficient in the real world. However, there was an outbreak of West Nile virus in Louisiana back like 2003, was almost certainly transmitted by Albopictus. They controlled all the Culex vectors as the outbreak started. Albopictus feeds on anything. Albopictus is an efficient vector of West Nile virus. But their control method was focused on spraying in the evening to kill the Culex, which didn't kill the Albopictus, which was a day feeder. So they killed all the Culex, which slowed the outbreak in the birds, but the Albopictus fed on the birds, fed on the people, and I'm sure the Albopictus was the principal vector for that outbreak. Remember, if the mosquito is active during the day, it sleeps at night, usually inside of a rodent burrow. So if you spray, you don't kill them. If you've got a nighttime feeder and you spray during the early part of the day, they're in a rodent burrow sleeping during the day so they can feed at night. You have to spray at the right time of day for that species. And yes, Albopictus are potentially dangerous for a couple of viruses. Next question. Anybody?
Can I throw something in here about virology?
Go for it.
Okay, you mentioned Trinidad. I don't know if you can see this book. It's called The Naturalist in Trinidad.
Yes.
And Brookworth was a virologist. He went all over basically the Western Hemisphere, studying arboviruses and trying to figure out in Trinidad why it was when they cut down the trees, they would get avian malaria or something. And it was because the mosquitoes were canopy mosquitoes. They didn't come down to where the humans were until they cut the trees down. And then people started getting this kind of, yellow fever. It was spread.
Yeah, this has been known for years for yellow fever.
Yeah.
When yellow fever came to the Americas, it was devastating. When Walter Reed showed that yellow fever was transmitted by Aedes aegypti, they virtually eradicated aegypti and eradicated yellow fever. However, there were monkeys in the jungle, you have a transmission cycle between monkeys and mosquitoes. Aegypti doesn't feed well on monkeys, but would infect a couple of them. There's some other mosquito species that feed on monkeys and are good vectors of yellow fever. But of course, the monkey's up in the top of the tree. This mosquito only lives in the top of the tree. Therefore, you had very few bites. What has been known for 50 some odd years is the woodcutter who goes out and cuts a tree. The tree falls down. The woodcutter walks along the tree, cutting off the branches. When the woodcutter gets up to what was the top of the tree, which is now 1 meter, 3 feet above the top of the ground, he's cutting wood. The mosquito thinks it's 15, 20 meters up in the sky and bites him because he's a monkey up in the top of the tree. That has been known for years. It's fun. Yes.
What's the status of the control of mosquitoes? Do they follow all of this stuff, or do they just go out and spray any time of the day, night, week, whatever?
Depends on who you're talking to and where you are. Most of the spray is, what is the principal pest mosquito? Kill it, which is what happened for West Nile and the Midwest. They killed a bunch of people because having the Aedes vexans there, which was their principal pest mosquito, regular control, they killed 95% of the vexans. They did as West Nile was coming across the U.S. in 2003. This mosquito control district said they were not doing a study, but they found 2 areas that were demographically identical. They flipped a coin and said, area A, we do regular ordinary control. Area B, we do super control. A month later, West Nile virus got there just as they predicted, and they checked cases every day. And then they panicked. they realized area A or area B with the super mosquito control had more than 3 times as many deaths per 1,000 people as area A. When they told me this, I said, "Oh yeah, you killed 80s Vexans." And he looked at me and said, "How did you know that?" I said, "Because 80s Vexans cannot transmit West Nile. It's an annoying, painful biter, and West Nile was in the paper every day about wear repellent, wear long sleeve shirts, et cetera, avoid mosquitoes." So the person in area A, where you killed 95%, there's still some Vexans. They got bit. They said, "Oh my God, West Nile, I'm going to die." And they put repellent on all over. And that evening, when the Tarsalis came out, they weren't being bit because they were wearing repellent. Area B, there were no Vexans because they killed them all. They said, "Well, those mosquitoes, they killed all the stinking mosquitoes. I don't need to wear no stinking DEET." The Tarsalis is a snake, it's a non-painful biter. So they got bit, they didn't notice it, so they died.
Oh,
How,
I didn't notice that.
so how do you, and depending on, oh, at this time I checked people in Florida to find out, I say collect mosquitoes and I will tell you whether they're good vectors for West Nile virus, yes or no. So I had various control districts collect mosquitoes for me, ship me live mosquitoes. I was more interested in Venezuelan equine encephalitis and Rift Valley fever. The 2 viruses I think are much more dangerous. So I tested them for West Nile, Venezuelan, and Rift, and then told them that Culex quincofasciatus and Culex nigropalpus are the 2 principal vectors of West Nile in Florida. And so, okay, they would focus their control on those species, which has been doing for 25 years. I said, if you had an outbreak of Venezuelan equine, which they knew about this virus, would kill those mosquitoes. I said, pointless. Those mosquitoes can't even get infected with Venezuelan equine virus. Venezuelan virus. It's Aedes teniorhynchus that is the principal vector for Venezuela. There was a recent, I was talking to a senior person, there was a recent series of floods, hurricanes in the south, and there was an outbreak of West Nile. Aedes teniorhynchus, the salt marsh mosquito, and it's just annoying, painful biter. And they said they purposely did not control them. They let them bite the people, they refused to do the control, or said they couldn't do the control. The reason being, this painful biter does not transmit West Nile. Therefore, people put on repellent and didn't get bit by the quinks and nigropalpus, and therefore didn't die. But if the outbreak had been Venezuelan, you've got to control it. You have to know this disease, it's this mosquito, that mosquito, it's that mosquito, and the control is spray day, spray night, spray salt marsh, spray around the house. It's all species specific, and it's different.
Pretty hard to make policy for something like that.
Oh, there is no, you cannot make a general policy. And part of this became, if you go back 125 years ago with Walter Reed, when they found out that these little containers produced the mosquito that killed people with what was yellow fever. And in Cuba, the US Army had just taken over the island. It was under martial law. They basically said, the US Army has the right to go to any house at any time. We are inspecting for mosquitoes. If we find a container that could hold water, you're going to be fined. I'm going to make up a number, 2 cents. If we found a container that actually had water in it, you'd be fined 5 cents. And if you have a container that has water in it and mosquito larvae in it, the first offense is 10 cents. The second offense, you're going to jail. And they went scattered all through Havana, checking houses. The word got out really quickly. These crazy Americans are coming here checking out. Make sure every coconut shell is upside down. Make sure every container is upside down, because you can't. 2 cents was real money, and they had sent a bunch of people to jail. Almost immediately, every house turned every outside container upside down because they didn't want to go to jail. Within about 3 months, yellow fever was eliminated, gone. You go to Florida and you try that. This is my private property. Do you have a search warrant to come on my property? You are not allowed on my property. You cannot inspect my property. And that's Key West. And that caused a dengue outbreak. It's because it's, how do we educate? Do I want the federal people to come on my property without asking? No. Do I want to have the disease? No. How do we educate people to say, when I was in Ecuador, I found in the jungle, went to a hut, and there was a whole bunch of coconut shells, all different, right side up, just thrown out there. And some of the coconut shells were sitting like this, filled with water, and had 100 to 200 larvae in each shell. And they're Aedes aegypti. This is the vector of, at that time, just dengue or yellow fever. And I pointed out to the homeowner that this was really dangerous, that these could transmit dengue. No esencudos, no, these aren't mosquitoes, these are worms. And I had my Ecuadorian friend explain to him these were baby mosquitoes, and next week these would be mosquitoes, and in 2 weeks he would have dengue. And he'd better go out there and turn every one of these cups. It took him 15 minutes and it was done. But how do you convince people to get the containers in their backyards, the tires, any container that can hold a bird bath? These are full of mosquitoes, mostly Aedes albopictus, but that is a bridge vector for West Nile. That is a really efficient vector for chikungunya, Zika, yellow fever, dengue, if then you don't have many dogs around. So how do we do this? I don't know.
Well, social Darwinism will help along with that.
Yes. I mean, I wanted to have the poster of a coconut shell filled with water, arrow, a child with microcephaly, a coconut shell upside down, arrow to a healthy little child. That's real. How do you get that message out to people? One of the things that we have sort of done is elementary school. Give a talk to an elementary school class on mosquitoes. Bring in larvae and rear mosquitoes from the egg to larva to pupa to adults and show them mosquitoes. Talk about disease. And as their homework assignment, they have to go to their home and they have to do a survey looking for containers that might hold water. It's amazing what a second grader can find. And then have them go back and now cell phones document it. Whoever, and then do you have larvae? Yes or no? Can you bring them here? We'll identify them. These kids forever will be turning every container upside down.